Afternoon, Bio 2 students. I'm going to be doing my last video for Unit 2. This is going to be wrapping up the Mitosis unit, Mitosis 2. Let's get right into it. So, um, earlier in my session, Salma, I drew the mitosis cycle. I kind of drew another one that emphasizes the M phase, which is mitosis. Mitosis itself is divided into 5 phases. Good way to remember it is PPMAT. So it starts with prophase, then prometaphase, metaphase, anaphase, and telophase. I would encourage you to draw them out. Uh, let's go over each phase. So prometaphase is when the condensed chromosomes finally become visible. It goes from that chromatin, loose genetic material, to the condensed chromosomes. The nuclear envelope begins to dissolve so that the chromosomes are just allowed to freely float around in their cells, and the spindle apparatus starts to form. So the spindle apparatus kind of looks like a little dot with a bunch of lines coming out of it. Those lines are going to be what pull the chromosomes apart during mitosis. So prophase, it's got this, and then it's got your chromosomes. It's the nuclear envelope, which is this part right here, is gonna dissolve, but it hasn't yet. It will once it gets to prometaphase. So, prometaphase, the chromosomes attached to the microtubules, those are the things that are coming out of the spindle apparatus. And they start to line up at the center. And then the nuclear envelope becomes dissolved. So, I'm gonna erase this and draw what the cell looks like during prometaphase. So, got the spindle apparatuses. I don't really know. Not an English major, I'm a science guy. That's prometaphase. Ooh, excuse me. Um, so a little bit more about spindle fibers. If you want to follow along in the slideshow, I'm on slide 5. The mitotic spindle is where that's what grabs the chromosomes. It's made of microtubules. Those are the actual like long spindly things that are doing the grabbing and attaching. And associated proteins. They control the chromosome movement during mitosis, and the origin is the centrosome. So that's like the little round part that the tubules come out of. Uh, Here on slide 6 is just a really in-depth drawing of a chromosome. It's good to know all those parts. So the centromere region of the chromosome, that's what attaches the two chromatids. At the center of each chromatid, those little green dots with the microtubules coming out of it, those are kinetochores. So that's what the microtubules grab onto to pull apart the chromosomes. Um, and then, yeah. Slide 7, I think this is. Yeah, it's just a more in-depth look about what happens during G2 prophase and prometaphase. I kind of drew that out already, but if you want to draw this slide, that is certainly going to help. So, up next, metaphase. So, during metaphase, the chromosomes are aligned at the metaphase plate, which is just the middle. Then at each end of the cell is your centrosomes with the spindle fibers. So in my example here, I have 3 dots. The big dot, that's the centromere that keeps the chromatids together. And then the little dots are the kinetochores, which the spindle fibers grab onto, like that. That's what metaphase looks like. The chromosomes are aligned at the metaphase plate, which is that middle. The microtubules pull at the kinetochores, but the cohesions keep the sister chromatids together. Okay, so centromeres, um, all the centromeres of the chromosomes are at the metaphase plate. So the centromeres are those big dots in the center of the chromosomes. Um, it's an imaginary structure at the midway point between the spindle's two poles. So it doesn't actually exist, but it's like that region where they are combined. So up next is anaphase. So in anaphase, the cell is kind of starting to divide. That your spindle fibers, they have separated the chromosomes like this. And where this little break, hypothetical break is, that was where the metaphase plate is. So the cohesions are released, or cohesions release the sister chromatids, and the sister chromatids separate. Microtubules are going to start pulling them towards the poles of each cell. Um, so the non-kinetochore microtubules from the opposite poles overlap and push against each other, and that's what elongates the cell like that. At the end of anaphase, the duplicate groups of chromosomes show up at the opposite ends of the elongated cell, so that's where opposite ends are right there. And then cytokinesis, which is when the cell gets cleaved into two, that's your C stage right there. Um, that usually begins during anaphase or the next stage, which is telophase. And the spindles here eventually disassemble. So, telophase, very similar to anaphase, except that the nuclear envelope starts to reform. So, I'm going to change to a different color for the nuclear envelope starting to reform. So, so telophase is the one in blue that I'm drawing. The um, the chromatids are much closer now. And it starts to regain the nuclear envelope, which is right here. Again, imaginary line right there, that was the metaphase plate. And that's where, um, so in telophase, the nuclear envelope begins to reform, the spindle apparatus is finally dissolved, and the chromosomes start to uncoil. So they go back to becoming loose genetic chromatin. And that ends the M phase of which is mitosis. Um, Here's on slide, what is that, 13 is another drawing of metaphase, anaphase, and then telophase and cytokinesis. If you want to draw those out, by all means, go ahead. Cytokinesis, cleavage of the cell into equal halves. So cytokinesis, that gets cut right there, and it becomes two cells full of that loose genetic material. And that's mitosis for you. Um, Here's the pull everywhere. And okay, so that's it for this video. I'm going to do one more from this PowerPoint, and I'll see you then.